Hi everyone. Welcome back to VRC Smart Little channel. Today I am with part 2 of biology, class 12 biology. Welcome back to Warriors batch, and this is chapter 4, principles of inheritance and variation. So if you have not watched my previous session, the link is provided in the description. You can go and check it out. In the previous session, we have covered about Mendel's law of inheritance of one gene, and we discussed about the various things about the law of dominance, law of segregation. So you can go and watch that, so you'll get a better clarity. So you can continue this video after watching that. Okay. So in today's video, in part 2, we are going to just see about cross pollination in pea plant, test cross, back cross, inheritance of two genes, and law of independent assortment. Okay, so let's quickly dive on to today's topics and cover it in a easiest way. Okay, right. So what is today's topic? The first topic that we're going to study today is cross pollination in pea. Okay, so in previous session itself we discussed that in pea plant both self pollination as well as cross pollination is possible, right? In previous class itself we discussed in pea plant both self pollination and cross pollination is possible. Why? Because pea plant is a bisexual flower. Pea plant is a bisexual flower. Already I told that both gynoecium and androecium, both the parts will be present in the pea plant. Okay, both female part as well as the male part, both will be present in the pea plant, so that both self pollination as well as cross pollination is possible in pea plant. Okay, so now we studied how self-pollination occurs. So when you get F1 progeny, when you cross it, you can easily have the self-pollination. If you leave the plant as it is, self-pollination will surely occur in the plant. But when you do cross-pollination, you need to be very careful while doing the cross-pollination. There are few steps that you have to follow while doing the cross-pollination. Yes, few steps you have to follow while doing the cross pollination. So let us see now how Mendel had done this cross pollination in case of pea plant. Okay, how he had done the cross pollination in case of pea plant. Let us see now. Okay, okay. So select a pea with contrasting characters. So you are going to select a pea plant. Okay, so you're going to take the flower of the pea plant. Okay, for example, you have white flower or you have violet flower. Any one flower you take from the pea plant. Okay, remove the anther. Emasculation. What is emasculation? Emasculation is nothing but you're just removing the male part of the flower. You're going to remove the anther part of the flower. That is the male part of the flower you are removing from the flower. That is called as emasculation. Okay, so both, I told it is a bisexual flower. I told both male and female parts are present. If both male and female parts are present, surely there is probability of that is self pollination and fertilization. The probability of fertilization is high. So we are removing the anther first. In the small age itself, before the plant matures, at that time itself we are going to remove the anther of the particular flower. Okay, so that is called as emasculation. Keep in mind, it's really important word. Okay, so you are doing it. Why you are doing it? To avoid the self pollination. Why we are doing it? To avoid the self pollination. So when you remove, see, it's a bisexual flower. Bisexual flower means both male and female part will be present. So if I am going to remove completely the male part, then only female part will be present. So you can call it as female plant, right? After removing the male part, only female will be just staying there. So if female is only staying there, then you can call it as female plant, right? Okay. Then collection of pollen grains from other plant. Collection of pollen grains. So after the female matures, you are going to go for another plant. Okay. Take another plant, whatever you need. That is from pea plant only. Another pea plant. You are going to take it. From there, you are going to collect the pollen grains. From there, you are going to collect the pollen grains. from the male parent. That is, you're going to collect the pollen grains and transfer it to the female parent. You are transferring it to the female parent. So surely when you transmit it to the female parent, and when male parent is crossed with female parent, surely what happens? Pollination will occur, and once pollination occurs, what happens? Directly fertilization will take place and the seed will be formed. So that produced seed you are going to collect, and you are going to get the new offspring from that. So this is a process by which you will do the cross pollination in case of pea plant. Clear? Hope you got a clear idea about that. One more thing, during this removal of anther, what they will do is they want to have the seed of their desired male plant. So only we are collecting the pollen grains from whatever the male plant that we need. So while after removing the anther, after the process of emasculation, they will do bagging. Why they are going to do this bagging means in order to prevent the undesired pollination. So they are doing this bagging to prevent the undesired pollination. So to avoid undesired pollination, they are doing the bagging technique. So they will cover the plant. So they will have the plant. So they are having the flower. Now after removing the anther, they will cover the flower, female part of the flower, that is complete flower, they will cover with paper bag. So that since it is paper bag only, no, so the air can go inside and come out. So this is done to avoid the undesired pollination due to any other agents. Clear? So that is what. This is another technique you have to remember. One is emasculation, another one is bagging. Clear? Right. Now I am moving to the next one. It is nothing but test cross. It is really easy and important concept. It is test cross. What is test cross? So test cross is made between F1 and F2 dominant phenotype plant and a recessive phenotype parent in order to know their genotypes of the dominant phenotype plant. So in order to know the genotype of the dominant phenotype plant in the sense, for example, the dominant is tall. So what is the genotype of this tall? You have to know. For that we are doing the test cross. For testing that we are doing the test cross. So what you will consider for the test crosses, you will consider the heterozygous dominant. Heterozygous dominant and homozygous recessive you will consider. Heterozygous dominant means already we studied what is the meaning of heterozygous. Right? Two different types of alleles will be present. Homozygous, same alleles. Okay, so different types of alleles will be present. Here same alleles will be present, right? Same alleles will be present. So let us do the cross now and just let us try to understand the complete information. Okay. Okay. So now let us try to do this. So in the test cross, first I am going to consider the see. We told that it is going to be heterozygous dominant. So I'm going to consider here the flowers. Okay, I'm going to consider here the flowers. What is the dominant character of the flower? In pea plant, dominant character of the flower is going to be violet and white. So violet flower, I'm going to consider it as capital W, white is small. So heterozygous dominant means it is going to be, here in this case heterozygous dominant is going to be capital W and small w. Homozygous recessive is going to be small w allele. Okay. So I'm going to cross here. Capital W, small w. It is going to be your violet flower. I'm going to cross it with white flower. So violet flower, it is heterozygous violet, it is homozygous white. So you are going to get the two different types of gametes here. This is parents, right? Parents, you are going to get the gametes. During the meiosis, you are going to get the gametes. One is going to be capital W and is going to be the small w. Here, small w, small w. You're going to get the two gametes here. So, what happens is, if I am going to put it in the Punnett square, once again I am recalling it, it is introduced by Reginald C. Punnett, right? So, it is important to know that. So, you are going to put it under the Punnett square. So, first I am going to represent the male, then I am going to represent the female, then you are going to have the capital W, small w, small w, small w. So what is the probability of getting here capital W, small w, small w, small w? Here capital W, small w, small w, small w. So if you see, means this is completely what? Violet flower. This is completely the white flower. So if you want to write the genotypic ratio as well as the phenotypic ratio, both is 2:2. Okay, both the ratio is 2:2. So if 2:2 is getting cancelled, what will be the ratio? The ratio will be 1:1. The ratio is 1:1. So whenever they ask the ratio of the test cross, this will be your sure answer. Always it is 1:1. Okay, whenever they ask the ratio of the test cross, always it is 1:1. So back cross. So back cross is nothing but, you can able to see the definition here, crossing the F1 hybrid with either of its parent. So F1 hybrid means you are getting the F1 generation, right? Whatever you are getting the F1 generation, that you will be crossing it with the either of its parent. That either of its parent should match with its genetical identity. Okay, for example, if your F1 hybrid is going to be the tall, then the parent who is matching with the genetical identity of tall, with them only you will cross. Okay, in the F2 generation, to obtain the F2 generation. So that is called as back cross. That is called as back cross. Clear? Now let us move to the next one. The next thing is, you can able to see here, inheritance of two gene, dihybrid cross. So now we are going to study about the dihybrid cross. Dihybrid cross means what? It is a cross between two parents differing in two pairs of contrasting characters. So monohybrid cross, we studied only about the crossing of one character, like we crossed tall plant into short plant. So only one character we have crossed it. But here we are going to cross two different contrasting characters. So round is one character, yellow is one character, wrinkled is one character. That is, these two belong to same contrasting characters, round and wrinkled. These two belongs to same contrasting character, yellow and green. So it is round yellow and wrinkled green. So round is one character, yellow is one character, right? So two characters we are going to cross it. So that is called as dihybrid cross. So let us first try to understand about the dihybrid cross. Finally, we will write the phenotypic ratio and genotypic ratio. Okay. So first, let us try to understand the cross. Okay. So first, I am going to consider the round yellow. Round yellow is a dominant character, and I am going to cross it with the wrinkled green. Wrinkled green. So I took round yellow and I am going to cross it with wrinkled green. Okay, so these two are parents. So now round yellow is going to produce gamete called capital R, capital Y. Wrinkled green is going to produce a gamete small r, small y. These two gametes. Now when they fuse together, what will be produced in F1 generation? Capital R, capital Y, small r, small y. This is the F1 generation. Right? This is your F1 generation. Now F1 generation will be self-pollinated. F1 generation will be self-pollinated. Let us see that. So your F1 generation is going to be self-pollinated. Then what you will get? So what is your F1 generation? Capital R, capital Y, small r, small y. This is going to be self-pollinated, means it is going to be having pollination with the same variety. Okay. So what are the types of gametes it can produce? It can produce 4 different kinds of gametes, right? So what are the things it can produce? Means it can produce capital R, capital Y. Okay, it can produce capital R, small y. It can produce small r, capital Y. Then it can produce small r, small y. Similarly, the same, this also can produce the same types, right? Capital R, capital Y. Then it can produce capital R, small y, small r, capital Y, small r, small y. So these are the gametes it can produce. Now, putting in the Punnett square, this side will be consisting of male and this side will be consisting of female. So what you will be getting here? What you will be getting here? I'm going to fill in the inside parts. Okay. So here you will be getting capital R, capital Y, capital R, small y. That are all you have to fill it here. Okay. 1 second. I'll use it. 1 minute. So again, capital R, capital Y, capital R, capital R, small y, capital R, small y. Okay. Then you will be having the small r, capital Y. Okay. Then 1 second. We are not supposed to fill here. Okay. Wait. So you are having 1 second. Wait. See, capital R, capital Y, capital R, small y, small r, capital Y, small r, small y. The same will be filled here also. So small r, capital Y, small r, small y. Now when these two combine, what you will get? Capital R, capital R, capital Y, capital Y, capital R, capital R, capital Y, small y, capital R, small r, capital Y, capital Y, capital R, small r, capital Y, small y. So this is the first set of combination. Next, what you will get? Capital R, capital R, capital Y, small y, capital R, small y, small y, capital R, small r, capital Y, small y, capital R, small r. Then you will be getting capital small y, small y. Then here you will be getting capital R, small r, capital Y, capital Y, capital R, small r, capital Y, small y, small r, capital Y, capital Y, small r, capital Y, small y. Then you will be getting capital R, small r, capital Y, small y, then capital R, small y, small y, then capital R, then small r, capital Y, small y, then small r, small y, small y. Now, if you just look after what is your phenotypic ratio, means that general appearance. So, general appearance, how would you choose here? You see, this is round yellow. Okay. So, I am writing the general appearance in the blue pen. So, the first case is round yellow. Okay, round yellow. So, this is round yellow. Round yellow. This is round yellow. Round yellow. This is round yellow. This is round yellow. This is round, but this is green. So it is round green. This is round yellow. This is round green. This is round yellow. This is round yellow. This is wrinkled yellow. Wrinkled yellow. This is wrinkled yellow. Okay. This is round yellow. This is round green. This is wrinkled yellow. So it is wrinkled green. So if you just count the number of round yellows, how many number of round yellows we are getting? So what are the types first we are getting here? First, what are the types we are getting? We are having round yellow. We are having, this is round yellow. Okay, this is round yellow. Second category we are having round green. Round green. Then we are having wrinkled yellow, wrinkled, wrinkled yellow. Then we are having the wrinkled green, right? Wrinkled green. 4 types we have. So round yellow, how many? 1, 2, 3, 4, 5, 6, 5, 6, 7, 8, 7, 8, 9. So your round yellow is 9 in number. Then your round green, how many round green are there? 1, 2, 3. 3 round green are there. How many wrinkled yellow? 1, 2, 3 wrinkled yellow we are having. And how many wrinkled green? 1. Only 1. So this is your phenotype. This is your phenotype. So whenever they ask you the phenotypic ratio, whenever they are going to ask the phenotypic ratio. So if I want to make it more clear, wait, I'll rub these things. So whenever they are going to ask your phenotypic ratio, phenotype, okay, phenotype. So I'll write this side. So you are going to get, this is 9, this is 3 you are getting, this is 3 you are getting, this is 1 you are getting. So whenever they ask your phenotypic ratio, how you write your phenotypic ratio? Your phenotypic ratio is 9 is to 3 is to 3 is to 1. This is your phenotypic ratio. Okay, this is really important to remember. This is your phenotypic ratio. Now let us come and identify the genotypic ratio. So your genotypic ratio I am writing in the green color. Just see. So I am just mentioning it in green color. This is the 1. Okay, this is 1. Only 1 is available. Okay. So other than this, no. Then the similarity, I'm going to mark it in different colors. So these two are similar, right? So these two are similar. So this is 2 and this is also 2. So like this you are having similarities. So this is 1, this is 2, and these two are having the similarity. Okay, these two. Okay, these three are having the similarity. These three are having the similarity. This is how you have to find the genotype, right? These three are going to have the similarity. This, and this one is having the similarity. Okay, also this one is also showing the same similarity. Okay, then these two, okay, showing the same similarity. Then small I, right? Small y. Then so like this they are showing some similarity. So I will give you one important trick in writing the genotype. You can easily remember the genotype. So first I will write, then you see whether you can understand the genotype or not. Then I will explain the trick. Okay, I am writing the genotype now. The genotype is 1 is to 2 is to 2 is to 4 is to 1 is to 2 is to 1 is to 2 is to 1. This is your genotypic ratio. So how could I tell it so fast? Because there is a trick. I will just teach you the trick. Now, 1 second you listen here. So first, instead of writing like this, you just remember. You see, you are going to write easy trick I am just teaching you. 1 is to 2 is to 3 is to 4. When you write like this, this is the order, right? Instead of 3, you are going to just write it as 2. Instead of 3, you just write it as 2. Okay. So 1 is to 2 is to 2 is to 4. Till here you are clear, right? 1, 2, 3, 4. Like that you, if you are writing means, instead of 3 you have to write it 2. So 1 is to 2 is to 2 is to 4 is to. Then 1 is to 2, again 1 is to 2 you have to write. 1 is to 2, again 1 is to 2. 2 times. Repeat it. Then finally 1. So this is very easy, no? You can remember it really very easy in this way. This is really easy manner to remember, right? So in this way, if you remember, you can easily write the genotypic ratio very easily. Okay. So this genotypic ratio is also important for you to remember. Okay. Right. So you remember it with this trick. Okay, 1 is to 2 is to 2 is to 4 is to 1 is to 2 is to 1 is to 2 is to 1. Clear? This is the genotypic ratio and this is the phenotypic ratio. Clear? Got that, right? Okay. Now, let's go for the next. So finally, we are here with Mendel's law of independent assortment. So Mendel's law of independent assortment is applicable for dihybrid cross. Dihybrid cross. It is applicable for dihybrid cross. What we are saying is, it states that when two or more pairs of triads are combined in a hybrid segregation. Okay, see, that is, you are having capital R, small r, capital Y, small y. So what you can able to see, two or more characters, two characters are involved, right, here. So one pair of character is independent over the other pair of characters. Okay, so if you see, means one, what is one pair of character here? Capital R, small r is one pair of character. Another pair of character is capital Y, small y. So in the formation of F2 generation, you can able to see that one character will not be dependent on the another. So here capital R small r is one character, capital Y small y is another character. This character will not be dependent on this character and this character will not be dependent on this character. So no dependency is there. That is what we called as law of independent assortment. And also new combinations, recombinations and characteristics are formed in the F2 generation. See, when you cross it, you have only the dominant characters. But when you see the F2 generation, in the previous slide also we have done, no? See this. In this case, in the F2 generation, this complete thing is what? This complete thing what you get is F2 generation. So in your F2 generation, what will happen? In your F2 generation, always you will get the what, you will get the some of the old combinations you will get, and you can also observe some new combinations you can observe in your F2 generation in case of dihybrid cross. So it is called as law of independent assortment. That is, one character will not be dependent upon the another character. They are independent of each other. That is called as law of independent assortment. One more thing, when it comes for law of independent assortment, you have, in this character you have to write capital R, small r. In this character you have to small y, capital Y, small y. So the probability or chances of getting the capital R in the gamete formation is 50% and small r is 50%. Similarly, capital Y is 50% and small y is 50%. And one more thing, you have four types of combinations, right? Here in this case, you have four types of combination. What is your capital Y, capital R, small? That is capital R, small y, capital R, capital Y, small r, capital Y, small r, small y. So you have four types of combinations out of this. Out of this four types of combination, out of 100%, there is a chances of getting 25% of the each combination. Okay, 25% of the each combination, there is probability of getting out of 100%. Out of 100%, you have the probability of getting 25% of the each combination. That is 1 by 4th. That is 1 by 4th of the each combination. Clear? Hope everything is clear for you. So thank you so much for watching this video, and if you have not watched the part 1 video, the link is provided in the description. You can go and check it out, and kindly like this video, subscribe to our channel and click the bell button for notification, and we will be upcoming with next few videos also. Stay tuned. Till then, bye.